our lunch in 5 minutes. And second, to those of you who have heard part of this story. Thank you very much, Phil, for this extensive introduction. I must say that Phil and Bob Warner have been my heroes in the studies of sex change, or I better say they are my rabbis. You know that religious Jews, when they have sex problems, they go to ask advice from the rabbi. And I was asking quite a bit of questions throughout my maturity from both of them. So thank you very much. I'll start with a short introduction on sexual modes in scleractinian corals. And as you can see from this outline, we can divide the two major modes. We have corals which are hermaphrodites and corals which are gonocores. Hermaphrodites, we have both sex, female and male sex organs in the same polyp or the same colony. Gonocores are different. We have male corals, female corals. Then we have broadcasting hermaphrodites, and which are divided into two. The vast majority are simultaneous hermaphrodites. Same thing we have with the gonocores, and to give some numbers into them, which are mainly taken from the recent review of Andrew Baird and Betty Willis on reproduction or sexual reproduction, you can see that the vast majority of corals are broadcasting hermaphrodites, about 72% of them, and much less are gonocores. But as you see, there are a lot of different ways corals are reproducing. And in fact, one of the secrets or one of the major reasons for the evolutionary success of scleractinian corals is their wide reproductive plasticity. I'll tell you the story of my story in terms of coral sex studies. I've been studying coral reproduction maybe for the last 30 years. And so am I and my students, and so are many students in Australia. And I think that one of the stories that I'm going to tell you, I think, is one of the most exciting stories that I came across throughout my career in coral reef research. It takes place in Okinawa. That's where most of my studies have been done on what I'm going to present. And the major disturbances in Sesoko, or around Sesoko, which is the place where I've been studying, are typhoons, bleaching, and sedimentation, which I'm going to present or show you, demonstrate in a few slides. So these are one example of the results of a typhoon. The typhoons are also, besides causing damage to reefs, they're very important in the sense that they lower the temperature of the water during summertime. So that way, bleaching is avoided. However, in 1998, this disastrous year for coral reef research, the water temperature in Okinawa in July and August was about 2 degrees over the normal or the average summer temperatures. There were no typhoons that year, and the result was bleaching all over Okinawa. About 80% of the scleractinian corals died. Almost 100% of the soft corals died. And if that's the way the reefs looked like before, that's the way they looked like during and after. So I was lucky to be there during all this time and record the changes. And we published them in a paper called The Winners and the Losers. So who are the winners and who are losers in corals? Unfortunately, until today, the reefs in Okinawa did not come to their former state. Species like Stylophora, Ceratopora, and Pocillopora are locally extinct. Poraitis species are clearly the winners. They're all over. And I'm introducing you this story very shortly just to tell you the big surprise that we came across later on. And this is the area of work. This is the island of Sesoko. This is mainland, Okinawa. Here is our main studies done. And this is the Sesoko Bridge. Those of you who have been around there, note this river that flows here and into the reef. And that's what happens after a typhoon, where you can see this mud or red silt that is brought to the reef. This is the bridge that I was talking about. That's the way it looks like after a typhoon, reminding you of what Malcolm had showed yesterday, very similar things. So the area of work, which is so beautiful here, down underwater is, which we call Fungiland, which you'll see in a minute why, is an area which has constant drainage of silt, of sediment. Visibility is about 2 meters. And we were extremely surprised to see hundreds of thousands of fungids in this particular place, where almost all other corals were dead. So I've never seen such a place before. This was in 2004. And of course, that called for studies on fungids, especially that almost nothing was done on their was known on their reproduction before. Very little was in the literature. And it was an ideal place to do such a study. In Eilat, for example, there are very few fungids. Eilat is a beautiful place, but all what we have is a reef of 3 kilometers, out of which 1.5 kilometers are a nature reserve, so there's not much left to study or to manipulate. And, but we are number one in number of publications on coral reef research. You're surprised. This is per square meter of a reef. So that's the story. Here's just to show you how dense are these populations of fungids. To start with, we were quite greedy, and we wanted to study all of them. So there are 12 species that you can see here, but it was impossible to do what we later on went through. And when I say we, it's Kazuhiko Sakai, who is my co-worker and colleague in Sesoko. So we concentrated on these 2 species, Ctenactis acinata and Fungia repanda, which will be the topic of my subject today. So the idea was to bring these corals to the laboratory. They're easy to get. You don't have to break them. They're single polyps. We brought about 400 of them, and we hardly touched the reef in an area that we didn't disturb later on. We tagged each one of these individuals, put them in individual aquaria. So throughout the study, which was 2004, 6, 7, 8, and 10 now, every year, we've been measuring them, weighing them, and of course, observing their reproduction. These aquaria were put in very large containers with running seawater, and just before the expected spawning, which we knew it should be around the full moon or so, we lowered the water, and then, as a matter of fact, if you come to Okinawa 5 nights before the full moon of July or August, look at your watches or dive around 10 o'clock, not around, at 10 o'clock at night, you will see the fungid starting to reproduce. They are very, very accurate. And more than that, we found later on that the same specimen reproduce exactly at the same time throughout the years. So they have an amazing internal clock that year after year after year, the same individuals reproduce at the same time. Here you can see a male during reproduction, a female shedding eggs, and this is the aquarium system that we worked on. You can see that very clearly there is a difference between aquaria with males and females. An aquarium with a male that you can see here is water which is opaque or cloudy, while aquarium with a female contains water which is like foamy-like, so there was no way that we could be wrong in terms of sexing these corals. Now we knew that, say, coral number 12 is a male and number 27 is a female. This was in 2004. We come in 2006, and we see that the one which was male turned out to be a female, and the female a male. So this was quite a surprise. We thought that this is a mistake. It can't be. And then a year later, when we increased the sample size, it's a matter of sample sizes, of course, the story was very clear that there was sex change, and not only sex change, but it was bidirectional. In other words, same specimen started as a male. In the consecutive year, it was a female, and then a male again. This was 2007. I can just tell you now the end of the story, which is that not only that they are bidirectional, but it's multiple sex change. Today, after last summer, we know that there are about 6 or 7 specimens that we have evidence that they've done it 4 times, male to female to male to female. So it's a goldmine in terms of the amount of information that we got on specific corals, and their length, their weight, their growth rate, their life history in terms of sex change, and so on. As you know, sex change is not a new story. It is well known in fish for many, many years, and this is an example of Pseudoanthias squamipennis that you can see here. It's a large male which monopolizes matings with many females, that you can see here, a harem. And that is, the male reproductive value is strongly dependent on its size. And in that case, natural selection will favor sex change from a female to male, which is protogeny. Why? We'll see later on. An opposite example, of course, is in the clownfish, in the Amphiprion, which is a monogamous system, male and a female together, and here the female reproductive value is more dependent on size, which is favoring a male to female sex change, or what we call protandry. The same situation occurs in corals where it is random mating, as we will see later on. Recently, in 2007, there was a paper published on Trima, a very small goby fish, which shows bidirectional sex change. And I think that was the first paper to show it in fish. But again, it's an old story also with plants or with trees, where bidirectional sex change has been shown in the issues plants that display labile sexuality in response to energetic or environmental constraints. Charnov put it in his well-known book on the theory of sex allocation. The theory or the model that he puts is that an increase in plant size and associated resource availability will maximize plant reproductive success or fitness through an increase in female expression. Now, as I said, there are lots of examples. One famous example is in orchids. If you take a male orchid, or if you take an orchid, put it in the shadow, it will become male. And if you take the same orchid and put it in good conditions, in sun, it will become a female. And the idea is that being a male is cheaper energetically than female. To develop eggs is much more expensive energetically than sperm, and that goes on also with plants. As I said, there are many examples to show that, where the principle is when conditions are good for plants, whether you have nutrients, water, optimal conditions, that's where you would expect them to be females. If conditions are hard, bad years, no water, they will become males. And just to show you some examples of the size distribution of the cores I've been working with is this photo. And we'll talk on the sex allocation theory, which will try to explain what is going on here. So the sex allocation theory is the way in which organisms allocate resources to male and female function. Part of it is the size advantage hypothesis that Bob has been pushing throughout his academic career, I think, and pushing also the idea that size matters, and I agree with you. So the theory predicts that sex change is favored when reproductive success or fitness increases more quickly with size or age in one sex than in the other. Here you can see some examples of the sizes of the very same coral, and to give you a perspective of size, this is only one polyp, okay? So they're really, really very big organisms. They come to about 60 or 70 centimeters, and this is one polyp, okay? Remember. So it's not a colony. Here is the review that both Phil and Bob put, which was my Bible for a long time. And again, putting the idea of the size advantage model for sex change, where it quotes or says that the direction of sex change, whether it is protandrous or protogynous, is determined by the relative reproductive success or fitness over the course of a lifetime of an organism for the two sexes. Remember, it's over the course of the lifetime, and that is why we can't really measure fitness unless you measure reproductive effort in the same organism through its lifetime. So it's a hard thing to do. And here's the model where you have on one side fitness or reproductive value. Here is the body size or age. This is the curve for the female fitness. This is the curve for the male fitness. If the female fitness is above that of the male, or it happens when their body size is small or they are young, there comes a certain stage when they are bigger or older where it is advantageous for the female to change sex and to turn into a male. That way, that female will increase its females or its reproductive value or its fitness. And that is the case where we have a sex change from a female to a male. Of course, the opposite happens when the reproductive value or the fitness of the male is above that of the female, and once they grow, there comes a stage where it is advantageous for the male to change into a female, where we have the case of male to female sex change, or what we call protandrous sex change. So the rule of thumb in both cases is to maximize the genetic contribution for the next generation. And that way, they increase their fitness. And this is a graph which summarizes, as a matter of fact, all the results that I want to show you. And we're dealing here with the two species, with Ctenactes and Fungi repanda, and these are allometric relationships of the two species between weight and length. And note the blue squares are females, red is males, blue, black are no reproduction, and this one are more added corals to the sample just to increase the sample size. So note that when they're very young or small, Ctenactes do not reproduce, they're black, or they're all males. They start their life as males, then come a point or a range where they change sex, and as I said, it is occurring already 4 times, and there comes a point in their life where they probably have enough energy and they remain as females. See this, there is only one male here, and probably was born as a male and remained male all his life. It's more interesting what is happening here. Again, when they're very young, they do not reproduce, or they are males. Here is when they change sex, and the interval here is much smaller than here. But the interesting point is when they're very old or much bigger, here there was no reproduction whatsoever for the last 4 years in these particular individuals. So the question is whether we have senescence in these corals. And remember, they're one polyps. And senescence in living organisms or in animals doesn't make much sense if you don't reproduce anymore. So what is really your role in evolution? I'm a grandfather already, so I do have still role in evolution. I can do babysitting to my grandchildren, but that's not in animals. So what is really their role is a question here. So we'll touch this later on. We published this story at the stage where we still had bidirectional sex change. And as I said, now it is multidirectional. So what is a big deal in sex change in corals if it has well been known before in animals and in plants? First thing is the first report on sequential hermaphroditism or true sex change in corals. It has been tried or looked at in different Porites species, which are gonocores, in Turbinaria, Pavona, Montastrea, but none of them has showed any sex change, and they kept their sex throughout time. And the second, of course, is the observation of a multiple sex change in consecutive years that these corals are showing. So as a matter of fact, we can modify now the model of Charnov, instead of looking like that, to look like that, where we have multiple sex changes throughout time. I have to rush. So I've been digging the literature, and unfortunately, I found this example of a multiple sex change, that there is a polychaete worm that shows the same thing of multiple sex change, where these are monogamous pairs. There is a small male and a large female, but the story here is that the growth rate of the male is faster than the female, and once it is bigger than the female, they change sex. So this goes to and fro, and they're doing it several times. I don't know how many, but definitely it's a multiple sex change. So the questions that we should ask now are, at this stage, is why change sex? And how do we explain the repetitive sex change that we have seen in Ctenactes? We divided the corals into 3 major sizes, like very large, intermediate sizes, and very small. And what we are suggesting is that at intermediate sizes, that's when they change sex, they cannot afford being females all the time. In other words, they don't, at intermediate sizes, they have insufficient energy to reproduce as female year after year after year. So their way to go around it is to change sex, to turn into males, and in the consecutive year, when they are males, they are recovering, if you like, or they gain some more energy to become a female the next year. And that is a system where they can maximize their reproductive value. So another question that we should ask are, what are the possible cues for sex change? Is it behavior? Probably not. In corals, they're not really behaving like fish, or they don't have any social pressures. Is it chemical cues like hormones? Probably yes. And for that we have done experiment where we married or we coupled corals together for a year. What we did was, as I say, to couple different sizes of these Ctenactes coral. In other words, as you will imagine, there are many variations that one can do that, the small with the large, different sexes and so on. But the ultimate coupling would be to have a large male with a small female. If you have a large male and a small female, both of them lose in the way if they stay like that. It's better for both of them to change sex because a small male can produce enough sperm to fertilize many eggs with a large female. So both of them are expected to change sex, and of course the control experiment will be the other way around. So that's what we have been putting down now. There are 50 pairs like that. We've studied it last year. It did show some promising results, but statistically it wasn't still there. As then I say, this is only there you can do that, and now we have 100 specimens which we have sexed them of 2 species, and we've coupled them for a year, and we are expecting to see what happens next year. Another point is that I've been talking all the time on weight and length, and the whole theory is just on the weight of the organisms, and we definitely want to know their age. So aging in this species is not easy. Some of them, you can turn them upside down, and you can see the growth rings below very clearly, and then you can determine the age. But most of them, you can't see these growth rings, and then you have to use other methodologies. To do X-rays or to do what we do with porites, coring and so on, you kill the animal. So we did use CT, or computerized tomography, and we've got beautiful bending, and then we could compare the ages that we determined out of the CT to the ages that we determined from our computations of the growth rings, of the growth rates of the different individuals throughout time. So we have great correlations now between weight, length, and real age of these organisms. I can tell you that the largest females are about 60 or 70 centimeters. They are, we know them personally. They produce very, very little eggs. So last year during summer, we were able simply to count the number of eggs and sperm per individuals. And what we got is a curve that looks like that. When you're very small, you're producing small number of eggs or sperm. As you grow, your eggs and sperm are more and more higher numbers. As you age, you go down. So this is one example that there may be senescence here. As you're becoming old, you're producing less and less eggs. Another evidence that there is senescence in these corals is by studying their telomeres. You know that the 2009 Nobel Prize winners were on telomere studies and studying the telomerase, which is an enzyme that adds up or repairs the damage which is done to the caps of chromosomes, because as we age, the number or the length of these telomeres is becoming shorter and shorter. So if in humans, for example, if when we are born, our telomere length is about that, when we are 35 is about that, when it is in my age, it's about that. So as we age, the telomeres, our telomeres are decreasing in their length, and that is not the case with the eggs and sperm, which here is where the telomerase, this enzyme, works, and they remain long all the time. So we did that with the corals, and indeed we found an important or significant negative correlation between the number of base pairs, the length of the telomeres, and the size of the corals or their age, a negative correlation, as you would expect with aging organisms. The sperm, on the other hand, was way, way higher. So this study is still in progress, and it's another way, as a matter of fact, to age corals without damaging them. So my time is up. Thank you very much for listening.
Yossi, thank you for a marvellous talk, particularly with the technical difficulties we've had. The computer has crashed, as you